Hi, hello. I'm Masahito Kawazu from National Cancer Center in Japan. Today, I will talk about the transcriptome analysis of breast cancer clinical specimens with long-read sequencer. There are many tumor types in which structure variations of chromosome are abundant. For example, there are many structure variations, SVs, in chromosome in a type of breast cancer. That is triple-negative breast cancer, TNBC. TNBC comprises 15 to 20% of all breast cancer and is defined by a lack of estrogen and progesterone receptor expression and the absence of RB2 gene amplification, which encodes human epidermal growth factor receptor 2, HER2. Whole genome sequencing of TNBC revealed abundant SVs that is caused by homologous recombination deficiency. Previously, we have reported the results of integrative whole genome sequence integrative genomic analysis of TNBCs in which whole genome sequencing and short read transcriptome sequencing were performed. As shown in this slide, SVs in the representative chromosome in TNBCs are shown by colored arches. And as you can see, the chromosome of TNBCs are heavily affected by various SVs. So what is the consequence of these various SVs? These rearranged genomic regions transcribed, and if they are rearranged, what is the characteristics of these of the transcripts derived from such rearranged genome? To address these issues, we started the analysis of full-length transcriptome analysis with IsoSeq using long-read sequencer SQANTI. When we started the analysis, the length of raw read was about 20 to 30 kilobase pairs long. And that is much shorter than the current state of art, and that exceeds 150 kilobase pair long. So we prepared the cDNA libraries for the IsoSeq analysis. cDNAs are generated by reverse transcription. The generated double-stranded cDNAs is ligated with hairpin DNA, adapter DNA oligos with hairpin structure. And starting from the hairpin loop, DNA polymerase goes through this circular structure repeatedly. Assuming that transcript is 2,000 base pair long and raw sequencing read is 20 kilobase pair long. In that case, it turns out that the transcript will be sequenced 10 times. That greatly improved the accuracy. And consensus sequence is called circular consensus sequence, CCS. So again, double-stranded DNA is made circular with hairpin adapters and sequenced repeatedly to generate accurate sequence information. In the isoseq analysis, CCS with similar sequence were clustered together to generate isoseq consensus trees that represent one transcripts. And as a whole, they comprise the transcriptome as shown in the right panel. I have to mention that we used isoseq2 algorithm when we analyzed these data. And isoseq2 is slightly different from the current isoseq3 algorithm, but the general principle is common to both. In this slide, the actual sequences of representative gene SOX9AS1 that we detected in this project are presented. SOX9AS1 is encoded next to and opposite of SOX9 gene. At the top of this slide, reference sequences registered in Gencode are presented. And as you can see in the middle, we detected various isoforms that spans SOX9AS1 and locus and the neighboring AC005152 genes. So we detected not only SOX9AS1 isoforms, but also the read-through transcripts. At the bottom, the actual sequences that we verified by Sanger sequencing of PCR amplicons are presented. So in this way, we have identified hundreds of thousands of isoforms that are not registered in Gencode. So the question is how to analyze these isoforms. In general, abnormalities in cancer genome are detected by comparing the detected sequences with reference sequences. In this case, however, there are no corresponding reference sequences to be compared with or mapped against. And of note, the novel isoforms that do not have corresponding isoforms are the most interesting targets of analysis. SQANTI is a useful tool to analyze isoseq trees. It classifies isoseq trees based on how they differ from reference sequences. For example, full splice match FSM isoforms are the isoforms in which splice junctions perfectly match with those of reference sequences. Incomplete splice match ISM isoforms are composed of non-splice junctions, but they do not have complete set of splice junctions. novel in catalog, NIC isoforms contains novel splice junctions, but they are composed of non-splicing donors and non-splicing acceptors. And in contrast, novel not in catalog, NNIC isoforms have novel splicing donors or novel splicing acceptors. And by using SQANTI, as shown in the right graph, many NIC or many NIC and NNIC isoforms were detected. But however, we had no idea how to proceed with the analysis after this classification. Now, ISO-seq3 algorithm implements some quantitative processes, but when we were analyzing these data, there are no suitable tools to quantitate ISO-seq reads. By the way, in general, long-read sequencing technologies are used for the construction of reference genome sequences of various organisms. So we consider that ISO-seq reads can be used as a reference. You can build a reference transcriptome using the information obtained from the samples to be analyzed from the target samples. And once the transcriptome is created, you can use that transcriptome to make comparison between samples or between subtypes. Also, you can make use of various analytical tools that are now available. So we created a pipeline in order to that can merge ISO-seq reads from multiple samples. At first, ISO-seq reads with similar sequences should be merged to reduce redundancy. By merging ISO-seq reads with similar sequences that were supposed to be derived from identical transcript, we can obtain catalogs of non-redundant transcripts for individual samples, and they were further combined to generate cohort-wide transcriptome, as shown in panel B. Once you have generated cohort-wide transcriptome is further subjected to SQANTI filtering to remove artificial transcript by random forest algorithm. Once now that you have a new transcriptome, you can evaluate, you can measure the number of reads, number of ISO-seq reads for individual ISO-seq reads. In addition, by mapping short-read sequencing data against the reference transcriptome, you can evaluate the expression of genes using algorithms developed for the analysis of short-read sequencing data. And for example, you can identify genes with differential expression or genes with differential transcript usage. And we named this pipeline as multi-sample long-read transcriptome assembly, MUSTA, and now it is available at GitHub. We evaluated the performance of differential transcript usage detection, DTU detection, with simulation data. We tested 5 conditions of NIC rate of 0.0, NIC ratio of 0.0, 0.25, 0.5, and 0.75, and 1. Even under the condition of NIC ratio of 0.0, that means all the DTU isoforms FSM, full splice match isoforms, and none of the DTU isoforms NIC isoforms. Even under that condition, performance of DTU detection with MUSTA-derived transcriptome is higher than that with MUSTA, higher than that with Gencode transcriptome. And as the NIC ratio increased, the performance of DTU detection with Gencode transcriptome decreased, as shown in the dashed line or black line. On the other hand, the performance of DTU detection with MUSTA-derived transcriptome remained constant, showed stable precision and recall. So these data indicate that you can evaluate the differential usage of isoforms, including isoforms not annotated, that is not annotated, by creating transcriptome using the information obtained from the target samples. This Venn diagram shows the representative data of DTU detection simulation with NIC ratio of 0.25. So with IsoSeq technologies and this MUSTA pipeline, we analyze the transcriptome of breast cancer specimens. In this project, 8 estrogen receptor positive 8 RNA samples from, the RNA samples obtained from 8 ER positive breast cancer and 14 triple negative breast cancer TNBCs were analyzed. And as a result, about 800,000 non-redundant isoforms were detected from 6 million consensus sequence. Of them, 300,000 isoforms passed SQANTI filtering, and 260,000 isoforms were unannotated. And 280,000 isoforms had multiple exons. We identified 3,000 unannotated multi-exon genes, and 41 of them were detected in multiple samples, and 10 of them were unannotated in Gencode version 28 that we used throughout this project. But they were newly annotated in Gencode version 34. Further, MUSTA transcriptome contains 17 translated but unannotated open reading frames identified in a previous report. So these data suggest that MUSTA transcriptome, MUSTA pipeline successfully captured the isoforms that are actually present but unannotated. To see if we used sufficient number of samples, we incremented the number of samples to be analyzed one by one. As shown in the left graph, although the graph did not reach a plateau, we detected consistent numbers of isoforms that were detected in more than 80% of the samples, as shown in green and yellow. So these data indicate that we were able to detect most of the essential isoforms. In contrast, a larger cohort will be required for the thorough investigations of the heterogeneity of the isoforms. The SQANTI algorithm classified the isoforms into 9 categories, as mentioned above. And again, I will explain the isoforms, categories, but FSM isoform is the full splice match isoforms, and NIC, novel in catalog isoforms, the isoforms that contains novel splicing splice junctions, but they are composed of non-splicing donors and splicing acceptors. And novel not in catalog isoforms have novel splicing donors or novel splicing acceptors. And we found NIC to have the highest number of isoforms, indicating that the pairing of splicing donor and splicing acceptors are far more diverse than registered in Gencode. And the second highest categories was NNIC, and most of them were detected only in one sample. But substantial numbers of NNIC were detected in multiple samples. About 2,700 isoforms were detected in all the samples, and most of them were categorized as FSM. Considering this heterogeneity, we classified isoforms into 4 categories. Isoforms detected in more than half of the samples in both subtypes were defined as unique, as shown in cyan in the graphs. Isoforms that were detected only in one subtype defined as specific, as shown in red and blue in the graphs. Isoforms that were detected only in one sample are defined as unique, as shown in dark gray in the graphs. And as shown in the right graph, the numbers of detected unique isoforms varied between samples depending on the total numbers of detected isoforms. In contrast, there was little variation in the numbers of common isoforms. So, assuming that subtype-specific isoforms may contain key molecules relevant to the breast cancer biology, we tried to detect differential transcript usage genes that use transcript differentially between subtypes. And indeed, we detected 455 DTU genes, including 46 cancer-related genes and 10 breast cancer-related genes, such as ESR1 and BCL11A. So we focused on TNS3 genes, tensine 3 genes. First, we confirmed that the novel isoform of tensine 3 is specifically expressed in TNBC, as shown in the black bars in the right graph, by reverse RT-qPCR experiments. As shown in the right panel, TNS3 protein contains phosphatase domain and C2 domains and SH2 domains, and reportedly contributes to cell migration, anchorage-independent growth, or metastasis in several types of cancers. And new isoform we found in this project is devoid of phosphatase domain and C2 domain. The novel short form tensine 3 is transcribed from novel first exon. The genomic sequences of the first exon is conserved among the vertebrates, and the peaks of chromatin modifications associated with promoter function or enhancer function were observed at the site of this first exon and in triple-negative breast cancer cell lines or normal mammary epithelium. But they were not observed in EL-positive breast cancer cell line. So these data strongly support the existence of this novel short form TNS3. To investigate the function of short form TNS3, we analyzed the gene expression profile of MCF10A cells into which viral vector expressing canonical TNS3 or short form TNS3 was introduced. And we found that TNS3 short form had impact on gene expression similar to canonical TNS3, but with greater effect size. And expression of about 3,500 genes were affected by short form TNS3, but of them, at least 900 genes were only affected by TNS3, short form TNS3. Gene ontology analysis revealed that several features associated with cell adhesions were affected by both short form TNS3 and canonical TNS3. But the effect size is again larger. The effect size of short form TNS3 is larger than that of canonical TNS3. As shown at the bottom, the formation of stress fiber of actin filament is enhanced in MCF10A cells expressing short form TNS3. And we also found that expression of Notch1, Notch3, and FGFR3 important drivers of breast cancer is enhanced in MCF10A cells expressing short-form TNS3. So these data suggest that short-form TNS3 has function distinct from canonical TNS3 and implies that TNS3 short form may contribute to the initiation or progression of breast cancer. We confirmed the expression of short form TNS3 in the cancer genome atlas cohort by calculating the percent spliced in of the novel first exon. As shown in the right panel, PSI is higher in basal type breast cancer compared to the other types of breast cancer. So at last, I will talk about the fusion transcripts. Using short-read whole genome sequencing, you can quite accurately and sensitively detect structure breakpoints of structure variations. On the other hand, by using long-read sequencer, you can accurately analyze the structure of fusion transcripts derived from such rearranged genome. Of the chimeric isoseq reads found in 9 TNBC samples, which were previously subjected to whole genome sequencing, we identified 400 reads with corresponding DNA breakpoints in whole genome sequencing data. By analyzing the context, genomic context, where the detected transcripts were encoded, we found that most 5' transcript fragments were encoded in the genic region. In contrast, many 3' transcript fragments were encoded in the intergenic region or non-genic region. Or we also found that most transcription start site will originate from canonical transcription start site. Also, we noticed that most 5' transcript fragments were multi-exonic, while many 3' transcript fragments were monoexonic. So in general, we can say that 5' transcript fragments tends to be regulated by canonical rules, while 3' transcript fragments tend to deviate from the canonical rules. We also detected 5 non-redundant reads that were mapped to 3 genomic regions and at the same time had corresponding breakpoints detected in whole genome sequencing data. Recently, pan-cancer analysis of whole genome consortium reported bridged fusion, which are mapped to 2 genomic regions that are connected by intervening genomic fragments. Double hop fusions we present here has internal genomic region of more than thousands of base pairs, and some fusions are even spliced in these regions. This type of fusion transcripts can't be found without long-read transcriptome sequencing. We confirmed the existence of double hop fusion involving ERVFLD1 by Sanger sequencing of PCR amplicons. Because the full coding sequence of ERVFLD1 is included in this fusion transcript, it is considered that the full-length ERVFLD1 protein was translated in this sample. And the expression of ERVFLD1 was exclusively detected in the sample carrying this fusion transcript. The expression of it was reported that expression of ERVFLD1 is generally suppressed across all the tissues except for placenta. Considering the chromatin modification status of the genomic loci, it is highly possible that the expression of this transcript is regulated by the cis regulatory element of histone 1H. The chromatin modification associated with promoter or enhancer function was detected in histone 1H cis regulatory region in triple negative breast cancer cell lines and in normal mammary epithelium. And these data suggested the existence of mechanisms like enhancer hijacking. Because it was reported that ERVFLD1 suppressed the anti-tumor immune response in allogenic mouse tumor model. So we measured the growth of tumors expressing ERVFLD1 in syngenic mouse model in which MC38 colon murine tumor cells derived from B6 mice was used. As shown in the left graph, MC38 cells expressing ERVFLD1 generated larger tumors than control MC38 cells infected with empty vector. In contrast, MC38 cells expressing ERVFLD1 and control MC38 cells generated tumors with similar size. So these data indicated that ERVFLD1 promoted tumor growth by inhibiting anti-tumor immune response of the host B6 mice, and that enhanced expression of ERVFLD1 in human cancer cells might contribute to the tumor growth. So in summary, we developed a pipeline for merge sample along with Transcriptome Assembly, MUSTA, to analyze isoseq data. MUSTA enabled construction of transcriptome from 22 breast cancer clinical specimens. And we identified subtypes specific isoforms of genes associated with breast cancer phenotype. And we also found functional double hop fusion transcript that was transcribed from complex structure rearrangement. So at last, I would like to thank many collaborators and members of Manolab. And I have to mention that major part of this work was done by Shinichi Namba, who joined our lab as undergraduate student, and now he is at Osaka University as a graduate student. So thank you for listening. And that's it.